Okay, wonderful. So this session is going to be focused on data struggles and challenges. There's a bit of feedback in the air. Give a second. So we've heard in a couple of places already yesterday and this morning about data processing pipelines that are being used. Let's try over here. Is that better? Yeah, okay. So we've heard in a couple of places already throughout the conference about some of the data processing pipelines that are in place, being used, and some of the struggles that are associated with them. But this is a very key aspect of everything that we do. And so it's a really important thing to call out. And so we decided to have a specific panel session focused explicitly on data struggles. So we don't necessarily want to hear about things that are working. We want to hear about the things that aren't. So have a think about that as you're considering what questions you might want to ask the panelists or what conversations we should be having as a group. So I'll go ahead and ask all of our panelists to introduce themselves. Let's start with Mark.
Hi, I'm Mark Basham, so I'm the Science Director for Artificial Intelligence and Informatics here at the Rosalind Franklin Institute.
Great, Samo.
Okay, my name is Samo Hudoklin. I'm coming from the University of Ljubljana in Slovenia. I'm part of the Institute for Cell Biology. And actually, I'm not full-time researcher, so I do in teaching. But during our work, we entered this field of volume EM and then data segmentation, and now I'm here.
And I'm Simone Weyand. I'm from the EMBL EBI, and I work at the Empire Archive, where we get all the raw data and make it available.
Okay, perfect. And I think the first thing that I want to start with, because we have a mixture of people up here across the life cycle of data, essentially. So the question that I want to start with is, what challenges do you think the field faces right now when it comes to data, data processing, and data archival? Does anyone want to jump in first, or should I call?
I can start, maybe.
Great.
So there are several different major problems. One problem is the huge amount of data that is basically included in one data set, so Empire was originally set up for single particle analysis. And if we compare the data set size, it's just a lot bigger for volume EM data and a lot more complex because there are multimodal entries which take a lot more time to annotate. So one challenge is to have the influx of data stable and that a lot of people can upload their huge data sets at the same time, which is, of course, a huge, it's a much bigger dimension than it was for cryo-EM initially. So to keep up with that, then also, of course, to harvest, let's say, the metadata and what metadata is needed that somebody else who wants to download and process that data set can process it without problems. That is another challenge because the metadata parameters that we need are very different from what we were used to. So I think this is the major challenge at this moment.
For me, so let's say that I would represent a typical scientist who are limited in resources, limited in time, limited in knowledge. And for us, let's say, bottleneck is that we don't have a tool that would be easy enough, accessible to do the real-time segmentations. Because if you want to, why we have this 3D data, of course, because we want to see some structures in 3D to get the function out of them. But then the next step with the volume EM is, of course, to do the quantification. And for that, we need segmentation of the data. And on one side, we have a lot of data, many volumes, many terabytes or even more. But on the other hand, there is a lack of annotated data in such a way that there will be some pipelines which will be unsupervised, that we can train the algorithms in a powerful way to get all the data we can throw in, because there is a huge variety of samples, protocols, preparation, everything that makes the total variety of the samples. And at the moment, we are still struggling with this.
So yeah, I think I completely agree with the other two panelists. I think volume of data is going to be a challenge as it is only getting larger, and all the new equipment that's being built is just making that a worse problem. But I think actually for me, the biggest challenge is all around actually collecting all of the metadata that's not associated with the instrument. It's the conversation that everybody had yesterday, I thought was fascinating, because there is very little standardized ways of capturing the additional information about, well, what cell was it? What prep did you use? Why are you doing the experiment in the first place? And all of those bits of information that are really critical for a lot of the way that we want to use the data. So we can do quite a lot with just data, but the segmentations, all of this additional metadata, none of that's really easily captured at the moment. I think that's going to be the biggest challenge for the field if you want to sort of go ahead and use some of the informatics or sort of data methods that you can potentially use on that data at a later point.
Great. Are there any challenges that are faced by the audience that you'd like to jump in to add to the conversation? I know we had a talk, I think earlier or yesterday perhaps, about registration of large volumes and flattening of large volumes. So I think that's potentially another area that we haven't spoken about yet. I saw a hand.
Well, we're recently started trying to do automated segmentation, for instance, of mitochondria in malaria parasites. And these are different, of course, from mitochondria in other species. So the problem we're facing is that there's all these things like MitoNet and other things where they have all kinds of data banks to train neural network type of segmentation systems. But as long as they're not either processed in the same way or from the same species, we're not going to be able to segment this because all the input data is just wrong. So we'll not recognize mitochondria in one way. So if we could filter all this data set saying, okay, this is at least the same resin and the same prep, so mitochondria will have probably comparable contrast. Let's start from there. That will already save us a lot of time in just getting frustrated with the fact that we can't train a neural network to actually do the segmentation for us.
Right. Okay. So linking the metadata to the data itself and the downstream processing.
Yeah. So you can actually say for all these data banks, make a selection. Say, okay, we're only interested in ones that are embedded in epoxy resin with osmium present, or we want actually to have the acrylic resin process samples. We're not interested in the chemically fixed samples because they will probably look different from the high pressure frozen sample that we have. Things like that. That would be, I think, a huge benefit in training neural networks to do the segmentation for you.
Yeah, great point. Go ahead.
I mean, yeah, okay. So when I said that there is no enough data, right? Of course, we can go deeper and to segment those things, right? At the moment, most of the protocols that actually, some learning protocols are biased, right? Or supervised. So that means that you have the data set, which is segmented and manual segmented, and then that is thrown to the pipeline. And then, of course, you evaluate the results on the same data set. So that means the results that were reported are pretty nice. But then again, we have to consider this complexity that you mentioned. Of course, mitochondria are one of the easiest organelles to segment, right? I mean, those still challenging, but the easiest if you compare to Golgi or ER or something more complex. But then you have to consider there are no standardized protocols. Each lab has its own way doing it that results in different contrast, different artifacts, different machines, all this. Then if you change the resolution or magnification, of course, most of those programs get completely lost. And then you have also the types of the samples. If you're doing with cell lines, which are fairly simple, right? In the cytoplasm, it's more or less some organelles, but empty space. I mean, empty. Of course, it's still complex, but comparing to some thermally differentiated cells that you can find in the tissues, that's a day and night. And it's all variety of shades. So all this, it's a problem. And if you want to, and then of course, time, you don't have the people in the lab who are specialized to do this. There are not many labs or that can afford to have a team to actually do the computer sciences and all this, right? And then you have many labs where people are like me. I'm also heavily involved in teaching. That means I don't have time to do all the things. But you have to be a core facility in one side. You have to be in the lecture room. You have to do the all kind of stuff. And then, of course, maybe you can do it, but you don't have time. So actually, time is also very much important here. Access to the machines, to the instruments. And actually, therefore, the colleagues that do the segmentations in our lab actually report that first step would be to get repositories, to get some consensus, to really find, to really define the data in a way, at least that you can find the data that you would like to. So to search all the data sets with mitochondria segmentation, right? But now you have to do it manually. There's no visualization tool. You have to download everything and then you find it's visible or not. So it's, I think there's first part of it.
Yes, and I think this again comes back to the metadata that we need to collect, because as opposed to other techniques in volume EM, it's very much dependent which programs you use, which parameters you use, and that dictates very much the result, how the result looks like. And if you get the same result as the original author gets, you need to know all these individual steps very clearly. You can't just put your data in a pipeline and then wait for the result. And I think this is one of the big challenges, to get enough metadata that everybody can more or less blindly reproduce the data. So very often we get people asking us, but this on this Empire entry, how did the authors do this and that step? Of course, we don't know. And I think this is much more crucial in volume EM, that we know exactly how samples have been prepared, how data has been processed, etc.
And just sort of go back to your point about sort of data processing, I think it's a really valid point. One of the things that I think we've had the most success with is where the experimental team has come to us really early in the process with what they want to segment, because that means we're able to actually discuss with them at that point, for example, which stains you might want to use, because we have example data like this that we could use for training. And obviously that's not always possible, but sometimes it is, and therefore it makes life a little bit easier. And I think actually it's a really important point when you're doing the experiments. You know, sometimes we get called in after the data's been collected and you're in the situation of like, well, I can't see the contrast on the thing you're trying to pull out. So no, there's really nothing we can do at this point. Whereas that conversation maybe a few months earlier would have potentially led to slightly different investigations in the first place. And I also wanted to build very slightly on, we talked about data volume, but that's actually becoming an increasingly large problem, because I'm just thinking of the skills that people in my team have that enable them to use supercomputers and these very, very large systems that are able to deal with terabytes of data. Talk earlier, you know, terabyte of data a day. I know EBIT creates vast quantities of data. Some of the volume EM techniques create huge quantities of data. And all of a sudden, that's no longer laptop. That's starting to not even be big machine in your lab. That's starting to be, you know, large scale compute. And using that is hard. And you need to have people who've got specialistic, you know, knowledge about that. You already have all of the knowledge about your experimental science, how to do your sample prep, how to collect the data in the first place. This is starting to add too big a load onto an individual person. So it really feels like collaboration with these large data sets is going to be key to all of the sort of the problems that you're going to address.
Just to feed into the how large a data set can be. So we had recently a data set from the volume EM community, which was 140 terabytes. That was only one data set. And then there were in parallel several 70, 80 terabyte data sets uploaded. So that is very normal.
Wait for a mic.
with this open science initiatives, which it's actually very good. But on the other hand, let's say we should, there are directions that we should store all the raw data, but what that actually means. So that each time you go to the microscope, that you store everything that you produced, then of course we will run out of the disk space in a week, right? But on the other hand, this is also like in our country, it's the funding agencies demand this, right? But on the other hand, they don't even have the personnel that could evaluate this, cannot check it. So it's really theory in a sense, right? So to get out what is meaningful to be offered and what is redundancy actually.
And that is also when people come to us and ask, what is actually raw data? How raw does the data need to be? Then usually for single particle analysis, we always say, well, as raw as possible, straight away the micrographs from the microscope. But having said that, for volume EM is a totally different dimension. So I think there needs to be an agreement what needs to be stored, what should be stored, maybe a bit of flexibility dependent on what method, etc. But that is also a very important point, actually.
I think going along with that, there's the idea of compression and what is acceptable to the community. So I think that's part of that same conversation. And then I just want to make one quick tie to something that Mark said about needing to be in communication with the experimentalists from the beginning. So that was the exact same point that was raised yesterday during the challenge panel that we had yesterday about sample preparation and how key it was to have those conversations with the researchers who have brought their sample in. And it sounds like that's also the case with the next step down, which is the data processing and data analysis. And it sounds like that isn't happening as frequently as it could. So that might also be kind of a takeaway from this. So I do think we had a couple of questions in the audience.
I do have a comment about organelle-specific, models. So you don't necessarily need to have lots of data from a specific organelle. So for example, MitoNet has been built on 1.5 million images EM. And it's been, of course, kind of focused on mitochondria. But in the lab, as I'm going to show you, I'll kind of talk after lunch, actually, we use it to segment every single organelle in our data sets, as long as there's enough contrast and the sample is done properly. So we use it for lipid droplets, and other things. So, of course, there are certain organelles that are difficult to segment anyway. And the segmentation, because it's starting from those 1.5 million images, is quite quick. We don't have to segment large-scale data sets. We can segment, generally 12 slices, just patches, not even the whole thing, just like little patches. And then the software does a pretty good job recognizing the organelles. It's not perfect, but it gets it right most of the time. So if you have mitochondria, which is different, from a different organism, if you use MitoNet, it's going to use the mito information from the software, but you can train on the structure of interest, can be anything, and then the software will recognize it.
So just before you have that microphone, can I just ask one or two questions? What level of knowledge and capability is required in that process, though? Because what you've just described is actually kind of an iterative process of, human plus the model to give out an output that is pretty good. And then I'm guessing that there's some proofreading after that. So what level of training was necessary to get someone from your team or yourself to the point where you can do that consistently?
It depends on the structure of interest, right? It all comes from the biology. If the structure is very homogeneous and it's very clear, like mitochondria are pretty obvious, on the kind of EM structure, as long as it's contrast. But then, learning how to kind of run the pipeline, you learn it once. And it took us a while to get there, but once we kind of do that, we have undergraduate students, grad students, it takes them like a week, to learn how to run models.
Okay, when you say it took a while to get there, though, can you just give me a number? Sorry to pin you down, but I think this is really important to discuss because a lot of times new software comes out and people think, oh, instantaneously, this will be available to me and it will work for me. And it actually does take some startup time in order to get to a point where it's useful.
Yeah, it's hard to quantify because, it's been a process, right? So I don't know if I had to start right now. And also we benefit from kind of personal relationship, people introducing us to the company and like kind of discussion, kind of resources, of course, but it's hard to kind of quantify it. But I would say that like the basic knowledge wouldn't take, kind of months just to learn how to Python.
Okay, great, thank you. We had a question here?
I have a question / comment on the raw data versus analyzed data deposition. So I've been discussing with people about like training new neural networks, specifically for cryo-volume EM, which is what the facility I work with mainly does. And like they've been asking to share the ground truth data from manual segmentation. And in the interest of like data findability and et cetera, I was wondering like what's the best way of making this ground truth segmentations public after they've been like published in journals, so that like people like Mark and the other people I've been talking with can reuse them to train new neural networks. And it's like somewhere that the data can be identified, can be scrutinized by the public, et cetera. There's no secret sauce. So what is the best way of doing this?
So experimental data can all go to Empire. Sometimes if you have a new entry type or for instance, segmented data, that is no problem either, but we might not have the modality yet. You just send an email along and say, I couldn't find this entry modality. Could you please add it to your spectrum of modalities? And then we do that.
Can I just clarify for a second, because I'm aware that the bioimage archive,
Yes,
I think,
it might
has
be-
a specific ability
Yeah.
to take segmentations and annotations.
Then probably it would go to the bioimage archive, yes.
Yeah.
So we are working very closely together and would usually internally then
Yeah.
say,
I don't think a full announcement has been made. I may have just stolen some thunder. But I do know that we were testing that functionality alongside of Matthew Hartley, I would say about a month and a half ago. So I think in the nearest future, hopefully there'll be a dedicated home for that type of thing. And I'll just, again, I don't want to steal thunder, but Elaine, if you want to wave, she has a talk this afternoon that is essentially a landscape analysis of where segmentations are currently deposited and why that is problematic. I'll let her say the rest later. Comments or questions from the left over here? Okay.
Hi, I was just wondering if we have a responsibility when we're designing projects with researchers at the beginning to dial back a little on some of their ambitions. I know ambition is great, and if you're maybe going into a new disease model or a new species, it's great to get the largest volume as possible to get lots of data, but it's not always necessary. And is it maybe a choice at that point, at the very beginning of projects, to just say, do you really need all of this? And maybe that makes everything a lot easier further down the line. I know people will always go against that and want bigger and bigger data sets, but should we be always asking that question at the beginning of projects?
Yeah, I mean, my personal thoughts on this is yes. Like we should be thinking about what's necessary and sufficient. And I think those are very key specific terms that should come up in conversations with people. And I think that it should be along the entire pipeline. So similar to what Mark had said, where we should be thinking about what is actually feasible. We're very good, I think, at doing this on the microscope side, right? Like, how long will it be until I can put that sample in? How long will it take me to collect the data given the current timings of my microscope and whatnot? But I think we're not very good at applying those same logical thoughts to the next step of if I've now collected, 100 terabytes of data or whatever it might be, how do I actually derive meaning from that data? What is my end goal and how long will that take me? I know that the proofreading has been mentioned, I think, once so far, and I want to come back to that maybe after some of these questions. But it's a common thing that's done that isn't necessarily always discussed or published. It hasn't been until recently. And if you think about the time that it takes, Mark, do you happen to remember that statistic off the top of your head from the recent publication?
Oh, so the Drosophila brain. So it was really nice because in the press releases for that, it highlighted the fact that it was 33 person years of proofreading time.
So that's not possible for most projects, clearly, right? So we should be having those conversations from the beginning to make sure that we actually are setting the researchers up for success in whatever it is that they're aiming for.
May
Go
I
ahead.
just,
Yes.
just small add-on. Actually, I don't think that we can avoid large data. Of course, on the individual basis, you can decide what is necessary for you, but actually you will get more and more bigger and bigger data. So that's for sure. And we are all there, right, with volume EM. But if you think about it, you have the same, you have even larger amount of data, or at least collections with the 2D dimensional microscopy, electron microscopy. And this whole field actually still needs segmentation, automatic segmentations. Because do you know how many, for instance, Golgis you have in your sample? What is the average number of mitochondria in your sample? Blah, 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 right? And if you want to answer some significant questions, then you have to compare this, let's say, normal condition with versus some pathological conditions. So you need some statistics, not only one sample, but multiple samples. So if you don't have a really good automatic segmentation, almost one button solution, right? You cannot afford to have learning curve of months for just one type of the mitochondria. So we need this. And for that, I think we should have an initiative, not only that someone comes to Jeff or to discuss this individually, but as a community. Mm-hmm. And for that, I think we should gather people who are doing this, what computer scientists need to solve the question. We have to introduce our, let's say, problems, what we want, how we do it, so pass all this knowledge to them, and then find, let's say, realistically a pathway which will lead to results. Mm-hmm. And that's why, because all of us, we have so different data, I don't think that the solution, okay, we have intermediate, let's say 5 to 10 years time where individual solutions will be taken, but then we need some unsupervised protocols that you can throw in all the data that you have on any scale, more or less, and get out some good results. And in a way, I was hoping that the Thermo Fisher and other producers will take this part, but they're also dependent on the data. So we as a community have to step in. And the bigger centers, networks, and so with much more funds and people and everything, will probably lead the way. But also we have to consider smaller institutes which don't have all this, at least money for paying things, but they have people trained to do something, some niche things, right? Everyone will do the mitochondria and those common things, but fusiform vesicles, which, for instance, rely only to the umbrella cells, to the cells that I'm studying, this is very unique, and no one will do this if there is no people in my place that can do this. So we have to make a balance between what is for the majority of the people, what we can use, and what is the specialty. So to keep everyone alive. Mm-hmm. And today, when we all know that it's hard to get good people, right? Technician, to hire a good technician now in our country, it's very problematic. And so we will see less and less people in the field of science, probably. Or at least we will have the moments between the good labs, or good financed labs and labs. So I think that we can organize meeting in this direction.
Don't look at me.
If I can just make one final comment on that point about the large data sets, I think you're right, the large data sets are kind of here to stay. I think definitely you can think about the problem, but people are always going to want to collect a full set of data, because going back to recollect is not always possible, or you get an amount of time booked on the microscope. What I would say, though, I think is so possibly underutilized is the sharing of that data. So it's a case of, once you've collected the data, you maybe look at the one thing you're interested in, and you've got a very small portion of your data that's actually used, and you keep the rest of it because you're going to look at it at some point in the future. And in 5, 6, 7, 8 years' time, it's still a set of hard drives that are sat on your desk or sat on your shelf somewhere. And maybe you find a PhD student that can look into it. But there's actually so much more information in there. And I do think that there's a big element of, you know, if you do collect a big set of data, that big set of data, just make it available after you've published your initial work on it, after you've published the thing you're really trying to do, so that the field can make use of it. And that can be a resource that everyone can access. So I think big data sets not always a bad thing, but big data sets sat on a shelf not doing anything useful is definitely a bad thing.
Just to go back to what Sama said about we need to organize a group who can move these things forward. I think Martin Wynn might have something to say about this, but also I think that we should be careful not to organize too many small groups that are all doing the same thing, because I do see that we are in danger of that in various places right now, especially with such a broad field as volume electron microscopy and all of the associated techniques that are linked to it that have, you know, sometimes their own larger communities also associated. And so we don't want to reinvent the wheel multiple times, because it will be slightly different every time we do it. And that is where there will be then long-term conflicts in terms of metadata structure and standards and things like that. So I do think the important thing there is we do need to have a set of groups that do it, but we need to have like one end output rather than multiple.
So like Europe bioimaging or some initiatives which cover those topics can, you know, gather people, and then we can directions.
Yes. Okay, I recognize there were a couple more hands in the audience just back here.
I'm wondering, for those of us that run shared resources and core facilities, we're all going to, you know, have a great time here, and then we're going to go home to our home institutions, and in many cases, we're serving a really broad community. We're enabling dozens of different workflows. Some are room temperature, some are cryo, some are volume, some are clem. And there will be a barrage of people pounding down the door who want to start the next project. And so, at least in my case, and I know a few cases for other colleagues, once we generate the data, we really don't have the bandwidth and time to help sort of nurture our trainees to go on to the next step. And I'm wondering if there are resources that we can point them to say, hey, you can tap into a pipeline here that will give suggestions of, okay, you need to start with alignment. These are different tools you can use. Then you need to move on to segmentation. Here are tools that you can use just to help some of our very newest people that are coming in. And I don't have the bandwidth or really even the expertise to point them in the right direction. So if I can point them to one place and say, go there and just read, and you'll find your next steps. I'm just curious on thoughts.
Yeah, no, that's a great question. And I think some people in the audience might have other thoughts, but I'll just, I think, hit some of the obvious ones. So there is a website called BII.edu. It might be 2 I's or 3 I's. I always forget. 3, thank you, Matt. And it is a lovely website that tries to list out all of the software that is available across a lot of different imaging modalities. And it also tries to tag them with appropriate tags. So if it only works on a certain system or type of system, if it's only useful for volume EM as opposed to cryo-ET or something along those lines, it will try to indicate that, as well as installation requirements and blah, blah, blah. Lots of information. It doesn't necessarily teach you how to use that software, but it is a good place to start if you're just trying to see what is out there and available. And then I would say image.sc, which is a forum that allows you to go and ask a question. And it's well attended, I think, by a lot of people in this audience and outside of this audience who will be able to answer some very basic questions. You may even find your question there already. They help you troubleshoot, they help you get started, they help you answer instruction type questions. And a lot of the developers are actually like active on that forum as well. So if you do run into bugs or need a new feature or something like that, you can usually talk directly to the developers on that forum. And then I would be remiss to not also shout out again the Slack channel. So this is EM of cells, tissues, and organs. So EM of CTO. And if you go to the Twitter handle for that, you can find the link to actually join the Slack channel. And there's a whole section on data and data processing and lots of friendly people there to ask questions. Are there any resources that I've missed that people want to shout out briefly before we other topic? Okay. Great. So I know we had a couple of other raised hands.
So microscopy technologies kind of are developing relatively fast. So how is it possible, or how can data analysis, data management, and algorithm development actually keep up with that? Also considering the fact that essentially many microscopy groups actually don't have that many data scientists, and then kind of data science essentially to develop methodology in itself kind of needs a certain critical mass. And the other question, which is related to that, what does this actually mean for standardization of methods, of data analysis, of data sets that are acquired with various instruments, which are all kind of somehow different? Okay.
Great question. On the panel, what do you think?
I mean, I can kick off with that one a little bit. I think it's a really valid point. I know, so when I used to work at Diamond Light Source, we had a similar problem that came up around sort of when buying new detectors for the beam lines, that you would spend $1 million or $2 million on a new detector, and what it would do is it would, change your data rates by an order of magnitude. And what wasn't considered in that process was all of the downstream. And it very quickly had to become part of the process of when you decide what new detector to buy, how are you going to deal with the networking, the storage? And I don't think it ever got as far as data processing necessarily, but I think that sort of holistic approach to thinking about the whole system, not just, when I buy this new instrument, what's it going to do? It's a case of, well, when I buy this new instrument, do I need a data scientist to go with it as well as somebody to run the instrument? I think is an interesting question that I don't know if anyone's currently asking in sort of groups, but I don't know whether that's the only approach, because essentially you're absolutely right. The competing requirements now are, climate change horrendous in terms of how much GPU you now have to throw at a problem to try and solve it in a way that requires only one person to do that. And the complexity is incredibly high as well, just for that sort of thing. So no, I don't think there's a solution, but I completely agree it's a problem.
And then in terms of like the standardization part of the question that you asked, I think we need to maybe look to other fields who've been through similar paths in the past. So not specifically the same data processing tasks that we're doing, but if you think about crystallography, for example, they had to completely develop a data processing pipeline 3-4 decades ago, right? And it's now completely automated. But when it wasn't completely automated, there were multiple solutions and multiple approaches and multiple packages to do it. The field got very, very large, and the pipelines got very, very large before it kind of standardized into very clear channels. And so I think we're in that growing phase, if you think about that shape of the path of travel as a field still. And I think it's a little bit difficult to sit in that space, but that's where we are. And I think we're hearing that in multiple ways, where we're saying we don't have enough data yet, we don't have enough annotations yet in order to do the automated processing, but we still are starting to be able to do it, but it's still really difficult. And so I think it's going to take time, and we're going to continue to grow. There's going to be a lot of options, and then we'll start shrinking down. Our microphone already out? Yes, thank you. Is it on? Oh, I think the battery might have gone then if it's flashing. I'll just do that. just pause. Anyone in the audience who's heard of SciCat, can you raise your hand? One? Yeah. Okay, great. Go ahead.
Yeah, so SciCat is a data catalog.
I thoroughly endorse it.
Yeah, I mean, I think we've heard enough comments that the functionality is nice and would be well adopted, but I don't think the community knows about SciCat yet. So perhaps that's a goal for us at the Franklin and maybe the SciCat community to do some outreach into the volume. But so.
I was going to say, I think it also brings up an interesting question because it's not the only metadata catalog.
Yeah.
There are plenty of other ones. I think most of the vendors have one that goes with their kit. What I think is missing is the interoperability of those systems so that you can easily actually relate them to one another rather than being sort of tied to one for one instrument and one for a different instrument. And it's that transferability, I think, is the key thing.
Yeah. Both of our microphones.
That's better.
Good. Okay.
So since Michelle name checked me earlier, I feel I have to comment on what we're thinking about at the moment. So there are moves afoot to try and put together a collaborative computational project for volume EM. And the idea of CCP is to provide support for software developments, computational projects within a scientific domain. And this has been done before for macromolecular crystallography and for cryo-EM and lots of other fields as well, and hopefully been fairly successful. So it kind of sits in the space between the microscopy, the data collection, and the long-term archives. So yeah, from the point of view of the facilities, you know, it's a body that can look at what you do with the data after you've collected the data. From the point of view of the archives, it's kind of representing the community, the people that are processing the data ready for deposition. And I think the general idea is both to help like the researchers, the practitioners. So how do you navigate your way through this plethora of tools and workflows? And also from the point of view of the developers, encouraging and helping them to work together so that you don't have, you're not always reinventing wheels. I'm conscious, you know, as Michelle's listed, there are lots of other initiatives in the volume EM field, and there's no, you know, are we just going to be yet another initiative? I think possibly the difference is that, yeah, it's more than a network. It's more than a exchange of ideas, but it would be a body that would actually look to do things, to jointly develop software, to bring software developers in, to come make solutions together and to kind of work closely with users and developers. So I just kind of want to highlight that this is something that might be coming as, yeah, hopefully addressing a lot of the problems that we've heard over the last half hour.
Yes, I have two comments. First is we're talking about interoperability, and I think file formats are very important about that. So I just wanted to drop OmiZar as a file format that is really great for storing volume EM data. Many tools support it already. Growing number of is adopting it shortly. So if you don't know how to store your large image files, then please use OmiZar and don't invent your own file format. The second comment that I want to make is an observation from the field of connectomics, which of course is a branch of volume EM. And their data analysis, there are now companies basically just doing that. They're specialized in that. So they will do anything from alignment, neuron segmentation, synapse detection. So no lab is, or a few labs are still doing that, but more and more labs are just outsourcing that because it's more economical. They don't have to find and train a personnel in their lab. And I think that is, as methods mature, is just one way of solving the hiring problem for skilled technicians and data engineers.
Can I just ask a quick question on that, because I'm not from the connectomics field? The work that is being done by the companies, that is still a semi-manual process. It's just being done through a company rather than us having to bring in skilled work in an individual lab, correct?
Well, it really depends on what kind of project it is and what the outcome is. But for doing like the things I was just listing, doing neuron segmentation, synapse detection, these are pretty robust and standardized routines now. So most of the parts are automated. And there is, as you said earlier, there's still a proofreading part attached to that. And that now makes up for basically like the biggest chunk of manual work. But there are methods underway under development to cut down on that. And that will dramatically fall in the next couple of years.
Just a comment about segmentation. I think it's necessary to have automatic segmentation tools for big volumes and huge volumes like half brain of Drosophila. But from experience, most of the volume data sets, they're not too big, like 100 gigabyte, maybe 200. And actually, you already have tools to extract all data from them by stereology. Volumes, surfaces, lengths, number of structures. You can do it without segmentation. The only thing you need to recognize the structure yourself. You just go through the volume and click on it. You get data. You don't need to segment in many cases, but not in all, of course.
Yeah, so that still requires the manual annotation to click on it, right? Yes, yeah, okay. So, but this goes back, I think, to some of the conversations that we were having about what is necessary and sufficient, right? Because if you can get your data analysis pipeline to work with that kind of less onerous pathway, then maybe that is sufficient rather than going through the full segmentation.
May I comment a little bit of last two questions? Comments. Actually, you can do it always manually. That's always an option, right? But it's not a really good option because of the time, at least, and the significance of the data. Because what is also problematic is that things that are biologically significant are not necessarily very broad, right? They can be some very small, let's say, branching of the mitochondria. Maybe that could be of interest, or some membrane contact sites or so something which is a bit hidden, and it's even harder to segment. Segmentation in connectomics is rather simple because you're finding just a line. So, but going to the ultrastructural level, it's completely another story. So I don't think that there will be very quickly solutions that can be just outsourced, because maybe you can have a manpower, like PhD students and so, but they can segment a lot, but they are not experts. So sometimes even the experts cannot really decide what is looking at, right? So to make it really precise, you need all players, so to say, and the field has to mature. At the moment, we have solutions which are partial. We have in the audience, people who can offer you to get a little bit further, but we are not quite there yet.
But I think you've just broached on a complete new topic that is really quite key and interesting, and that is bias and uncertainty, right? Because if there is uncertainty between experts and we're using expert annotation as our training for our machine learning, deep learning models, then how do we trust our deep learning models? I think there's like a whole thread there that we need to maybe think a little bit more deeply about.
And then we also have different modalities. So at the very end, we don't want just to have annotation of one volume, but we would like to combine light microscopy data, genomics, volume EM, all this to make a more robust story. So also this has to be, in a way, think about to prepare the field for annotations.
I think we have a couple of comments.
I think an important concept to bring to this discussion is the difference between semantic and instant segmentation.
Yeah.
So if we collect data and we do semantic segmentation, that's the one that was like discussed just before, we can kind of provide information about cell content in terms of overall volume. So we can say that cell is like 25% mitochondria, 10% ER, and so on and so forth, right? But what we're really going after is understanding how many of those mitochondria, what the distribution is, how many content make with other organelles. That's only achievable when we do instant segmentation. So we not categorize a class of objects, but we identify the individual objects within that class as specific. That requires a set of analysis that is different, tissue intensive. So that's a different ballpark. Right?
Absolutely. And that again goes back to the what is required by the project in hand.
Yeah, I'm having kind of finished my plug. I wanted to ask a real question. Actually, I want to go back to what Mark said earlier about hard disks languishing on shelves. I mean, do we feel that we've won the argument that data sets should be shared publicly or not? And I'm kind of coming from the macromolecular crystallography background, which is often touted as the perfect example of scientists working together for the common good, resulting in AlphaFold. But I think the truth is that it was a struggle over 30, 40 years to slowly extract data out of the grasping hands of the crystallographers. So I'm just wondering where the volume EM field is at the moment.
So are a large group, but we aren't all of the people in the field. So let's do a quick poll of the entire audience. Do you think that all of your data should always be submitted to a repository? Raise your hand. Okay. Definitely not everyone. So maybe 15 to 20%. I'm going to ask one or two more. Hold on. Yes. Yeah. Agreed. So then let's say the opposite. So I think none of my data should ever be submitted to a repository. Agree with that statement? Okay. So I think we're in between still. If I can, we're going to parse this a little bit more during the Mentimeter survey tomorrow, which will include everyone in the room, but also be hybrid with people online as part of the town hall. And we'll be sharing those results with the entire community afterwards. So hopefully we'll be able to get a little bit more fine detail than what I've just done. Dan.
Yeah, I just wanted to, there's been a lot of talk about segmentation, this segmentation, that. Do we actually have any idea of how reliable any of this segmentation is? I mean, it's going back to your point, and I remember the conversation I've had with you and Mark about, everybody's all crazy about AI and will do all of this, but, if you're feeding your trained models with crap, then, well, they're not going to get better.
Yeah.
So, where are people at with kind of assessing the reliability of the segmentation, and do we have a reliable metric that then we can get some confidence from and then start doing?
Yeah. So I think the standard right now in the field is the DICE coefficient, which is just a voxel correlation between like what has been done manually by an expert and what was output by the machine.
You've mentioned manually by an expert. Now, I'm sure you put 10 experts in a room and ask them to segment something and you'll get 10 different answers.
And that's exactly where I was going. Fine. You're passionate. So yeah, there's clear variation between, experts when they annotate. And I think we haven't, as a field, yet done our due diligence to understand the variation from the experts and then take that through to an uncertainty or an understanding of robustness or reproducibility from the perspective of what the output is when trained on that variation from the expert. So that is absolutely something that we as a field should be thinking about, in my opinion.
So you need a fudge factor, a B factor, which is used everywhere and anywhere. Yeah.
Yeah, I mean, it may not matter, right? So if you think of this, goes back again. I think a lot of this is linked to what is necessary and sufficient, right? Because if you're doing a comparative analysis between a pathology and like a wild type or a normal outcome, you don't necessarily need actual numbers. You don't need to say this has 5 mitochondria and this has 10 mitochondria. You just need to know the relative difference, right, given the situation. So I think that there are some ways that you could standardize against a wild type or a control in some way that would remove some of the variability there because you're not actually going for the full definition.
I don't know if biology is variable by, I mean, I started out as doing structural biology and then I ended up doing some volume EM and doing comparisons between wild type and mutants for different things. And yeah, not everything. It's so variable that you need a lot of numbers to.
Yes, yeah, absolutely. You will need lots of repeats. I agree.
I think one of the places though where there's potentially some nice opportunities is in all of the coincidence and correlative imaging, where you can use one modality to provide you with ground truth, or at least information for the other modality. And so there's some potentially nice places there, but I think it's the main problem at the moment is the resolutions are a mismatch, but there's a lot of work going on in the technology in those areas to improve that. So there's possibly some sort of low-hanging fruit that may come up in the future with interesting sort of comparative studies.
Thank you. So, not related to this, going back to the previous point about the data deposition. I am all for data deposition, but as a facility, the scientists don't always agree with it. So I think maybe we need a sort of consensus from not just us, but from maybe the journals that there is a requirement for them to deposit their data in an open source repository as well.
Yeah, that's a great point. And so the facility management panel will be tomorrow, and I think that this is something that we should discuss in more detail then. Is there any other burning questions, comments? Because we have about 2 or 3 more minutes.
Hi, yes, I really want to talk about quality of data that gets uploaded. As a software developer, I want my tools to work on everyday data. So therefore, we need everyday data to be available fairly. So it's a plug to please put your data that didn't make it into your Nature paper online as well, because that's really helpful for us.
Yeah, and that ties back into the question that was asked earlier by Rob about all data. I think that is a key point because AI learns what it is taught. And if AI has not seen poor data, then it's not going to know what to do with it. Even if all it's doing with it is excluding it, it's still not going to know what to do with it. Oh, sorry, Dimitri.
Yeah, I have a question. My question is if when automatic segmentation is applied, if anyone is also considering to also store the information before there is a thresholding, meaning when a classifier or system that segments decides to a particular class, it may be uncertain and very hesitantly assigned it to one of the classes, or it will be very, very certain that it's one class. So my question is if apart from the classes, the confidence information is being stored, if anyone is also storing this information.
Mark, do you want to?
So I know we're looking at trying to find nice ways of storing that information, but I think currently there's a lot of ways of storing that information, but I think the problem would be the size, because essentially by the time you've got your raw data and you've got your segmented data, and then you're duplicating it up potentially 4 or 5 or 6 times based on the percentage chances of all of the different classes, I imagine Empire would not like that.
I mean,
Yeah, I think this is the important point where we need to agree what data is sufficient, that can be uploaded, should be uploaded, and what data doesn't necessarily need to be uploaded.
So I think it's relevant and useful information to have
Of course.
for being able to judge
Yeah.
these things, but there's other, yeah, it's the purely a science thing.
We take all these big data sets, and so far it works really well. So we are aware that a lot more data is coming as the field grows. So yeah.
Okay, we're gonna have one more and then we need to wrap up for the session. So.
Yeah, so I heard the phrase poor quality data and thought I'd put my hand up. I guess it's kind of linking a few of these things. So I collected a large block-based data set, which was necessary and sufficient only. So this was essentially, the question was, you know, identifying tissue versus not, so just segmenting sort of the architecture. They've seen the data set, great. Now they see, oh, can just about see the nuclei. We want to segment those, and we're talking, you know, hundreds per section, you know, 2,000 sections. It's huge, and I guess it's kind of, you were talking about it just then anyway, like how poor quality is too poor quality to even try in terms of, you know, the cost, the time, difficulty. Do we just go back to, you know, putting more metal in long acquisitions? And maybe that is probably the best thing for this kind of data set. And that's not even worth uploading or attempting to, yeah, try and do analysis on this.
Yeah, that's a great question. I think, like, if it's a purely bespoke situation that you don't anticipate ever happening again, then it's maybe not worth putting the effort into somehow automating the processing, unless it's very precious. And if it's something that you anticipate doing again and again and again, then it's probably worth putting in the effort. And I think that goes broadly across the community. So if we see lots of facilities or lots of users doing similar things, then as a community. That's my opinion. Does anyone else want to add anything?
I think the only point I'd add to it is, I think it's an area where I've definitely thought about in the past but never done anything about, is the cost of ownership of data and the cost that, so for example, when you're storing data long term, would it actually be better to ditch that data and recollect it? Because that's a cheaper way of doing it, and it's going to be more effective long term. I think the same is true with data analysis. It's a case of it, you should be able to break that down into a purely cost-based thing. Is it cheaper to collect the data again and get better quality data, if that's possible? Or is it cheaper to try and analyze the, you say, poor quality data? But I think those are difficult cost estimates to make. And I think there's an awful lot of hidden costs, especially in data processing, that people aren't always thinking of. So.
Okay, thank you everyone. That was really fun. And I think we covered a lot of information, a lot of different topics together. So some areas I think we discussed, and maybe there are some actions that we can take as a community, is we need to be depositing our data. I think that's raw data as well as the annotations and segmentations that we create. And we need to, as a community, I think, come together around some metadata standards that describe that data appropriately for reuse. And I know that that is not a simple thing. So I think that will take some time and some effort from a large number of people, because we want there to be one cohesive standard rather than multiple standards that are competing. We also discussed, and I think this is a trend throughout the conference, that we need to be having conversations about the entire pipeline rather than just pieces of the pipeline, especially when we are starting projects, because the downstream implications can be addressed as part of those conversations.